Hello. Clear my voice?
Hello.
Hello.
Yes, sir. It is audible.
Okay. Okay. Right. So today we discuss the. Today we discuss the structure of the ovule and development of the female gametophyte. Is it audible? Okay. Hello. Please respond.
Yes, sir. It is audible.
Okay. Okay. Now. Okay. What is the ovule here? So means here the structure of the ovule and the development of the female gametophyte here means the megasporangium along with the protective course that we call as the integments, which is known as the ovule. So including the integment and the megasporangium we call as the ovule. So after the fertilization, these ovule form as the seed. So that's why the megasporangium is very important for the understanding the structure of the seed. So the first here the Grieve and Malfigi study the ontogeny of the young ovules and developing the seeds. So they describe the most conspicuous parts of the ovules. So after that, the brown and the mirabule, they are also describe the structure of the ovule. Okay. So the ovule is generally consists of the nucellus and enveloped by the one or two integments are there. Okay. Anything chemical क्या था? Chemical हाँ? क्या क्या है? क्या दिखा रहे? क्या है? इतना ना? Minimal का इतना देगा नहीं इतनी का हाँ. Starting कर देते काले का. Sorry, okay. Actually, Ibrahim. So the nucellus and enveloped by the one or two pigments are found. So it is found to attach to the placenta. And the inner wall of the ovary are by stalk known as the funicle.
మనం సాగర్ రింగ్ రోడ్ ఉంది కదమ్మా మెయిన్ రోడ్ లో పోవాలా బిఎన్ రెడ్డి దాని తర్వాత రైట్ సైడ్ గుర్రంగూడ దగ్గర రైట్ పోవాలా
ఎగ్జాక్ట్లీ సార్ చెప్పింది ఫాలో కండి రిటర్న్ తీసుకుంటే హాయిగా అక్కడ వెళ్ళిపోవచ్చు మళ్ళీ నార్మల్ ఫ్లైఓవర్ అనుకున్నాను The inner wall of the ovary by a stalk known as the funicle. So the part of the funicle that's run the parallel with the nucellus called the raphe. An ovule ready for the fertilization. It consists of the nucellus enveloped by the two or either one integments are there. Okay. So here, this is the one opening part is there. That one we call as the micropyle, and this is main passes for the pollen tube into the ovule. So the chala is the region where the integments are inserted. So the funicle and raphe, chalaja, and nucellus form the continuous tissue. The central axis of the ovule and the no sharp demarcation can be drawn between these parts. Okay. So next one is the types of ovule. So here in the angiosperms, mainly 6 types of the ovules we are recognized. So based on the position of the micropyle and the funicle, based on these position, we are described or classified the ovules into 6 types in the angiosperms. So the first one is here arthrotrope. Here, see here. Okay. So first one is the arthotropous ovule. So in this one, the micropyle and funicle lays a straight line. So see the first diagram, that is the first one. This is the micropyle and the funicle, and chalaza is also. There at least a straight line is there. This type of the ovule seen about around the 20 families. So mainly Polygonaceae and Piperaceae, we can see this type of arthropous ovule. Okay, arthropous means micropyle, funicle, and chalaza. They are placed a single line or straight line. The next one is the anatropous ovule. So in this one, the ovules become completely inverted, and as the result, which is the micropyle and funicle is closely each other, very close to each other. Micropyle and funicle means the ovule completely inverted. So that's why, due to these reasons, the micropyle and funicle very close to each other. So this type of the anatropous found in the among the angiosperms, 204 families are there, around the 200, 100 above, 100, 200 above families. Most of the families are in anatropous ovules are found. The next one is the hemianatropous. So hemianatropous, here the funicle lives at the right angle to the nucellus. What happened here? The funicle is lays to the right angle to the nucellus, means the ovule body. So it is the around the 13 families, means 10 to 13 families. We are see this type of the hemianatropous ovule. Okay. So next one is the comphylotrophus. So here in comphylotrophus, the ovule will be curved. Ovule, what happen here? They can modify a small curve-like structure in the comphylotrophus. So here, these Compylotropous ovule, we can see the Chenopodiaceae and Caparaceae. And fifth type of the ovule here, Amphitropous ovule. So here, Amphitropous ovule. So the ovule became a horseshoe shaped. The oval is totally curved to become a horseshoe shaped structure we can see in this type of the oval. Okay, so the curvature of the oval is more pronounced here. The curvature is also extend to the embryo sac became the horseshoe shaped structure you can see in this one. Here Amaranthesi and Butamaceae families you can see the Amphitropous oval. So the last one, there is the sixth type of the ovule here, Sarcinotropous ovule. So here, Sarcinotropous ovule is very peculiar type of the ovule, which leaves the panicle is exponentially long to form a complete circle around the ovule. So it is resemble to the another integument, means to form a another type of the integument, a complete circle around the ovule. Okay, so but it is free from the except to a small area of the attachment at the end of the funicle. Okay, it is free at the end of the funicle. So complete circle, the funicle is very long and to modified as a circular structure. That's why we call as a Sarcinotropous ovule. So in the mostly occurred in the Plumbagiaceae family and Cactiaceae families also. These two families are you can see the Sarcinotropous ovule. The next one is the integments. So the integments, the angiosperms, the ovules is consist of two or sometimes no integments also there. So that's why we call as a uni integument by integument or a, no integument. So uni integument means only the single protected layer is there. Then by integments means two integument layers are there. Then another one is the A integument. Integument. So here, okay. So here some extra structures are there on the these embryo sac. What is that one is the aerial and areolode and sarcoptesta. So these structures are associated with the integument. Additional structures are there. These are attached to the integments. So the Maheshwari in 1950 is defined as a new structure that arises from the base of the ovule and form a third, uh, which is called as a third integument. So aerial is we are considered as a third integument, not a integument like, but considered as a third integument. So it is the secondary collar like outer growth which is developed from the funicle and surrounds the ovule almost completely by the fertilization stage. Okay. So next one is the arylode. So here arylode, these Higel in 1957 The visual is recorded the presence of arylode in the Saphendaceae family, which is an outer growth arising from the tip of the outer integument grows downwards and covers the seed either completely or in a part. So this can be also regarded as the exclusively developed carancule. So carancule is also another type of the additional development. The next one is the sarcotesta. So the sarcotesta is the outer integument. Sometimes it became a juicy or fleshy structure is there. That one we call as the sarcotesta. It is often the pigmented and rich pads in the some members of the Mangueliaceae family. The sarcotesta is a fleshy development. Growth means additional growth. These structures, namely aryl and arylode sarcotesta, are attractive to the animals like birds and mammals and insects, other insects also, which are helped in the seed dispersals. These are the additional developments, so that's why it is the attract the animals, insects, mainly help in the means seed dispersal. Okay. So another one is the endothelium. In the several tags of the gamma petale, so mostly integument, and that one we call as the tenunucleate ovule. Means one integument is there, and tenunucleate ovules are seen in the gamma petale order. So cells of the inner epidermis of the integument is elongated, and aqueous dense cytoplasm is present and develop the conspicuous nucleus and differentiate into endothelium. So the inner side of the epidermis of the integument to modified and dense cytoplasm is there, that we call as the endothelium. In the most cases of the endothelium is differentiated at the time of the megaspore tetrad formation. So as the ovule grows, the nucellar epidermis is degenerated and the embryo cell comes into direct contact with the endothelium. So here the endothelium is generally single layered and the cells are uninucleate cells are there. Some are the multiseriate and multinucleate endothelium also observed in some members of the Asteraceae. Majorly uninucleate cells are there, but in multinucleate and multiseriate observed in the Asteraceae also. In the pedicularis, the endothelial cells became polyid condition is also there. The multinucleate condition of the endothelium also occurred in the balanitis. The endothelium normally surrounds the entire embryo sac. It may be restricted either the micropolar or talagel end of the embryo sac. It is usually degenerated after fertilization, but some families are persistent to the seed development stages. Okay. So here, the structure of the endothelium cells, we can see secretory of the meristematic cell. They are rich in the RNA and protein and some enzymes also there. So The functions of the endothelium are variously interpreted. So, basing on the location of the cytological features, many believe the serves the function of the nutrient like the anther tapetum. So, mainly to produce the nutrition for the megasporangia. So, operculum. So here operculum. So in the Hegel mirror in 1869, there was recorded the presence of topper-like structure on the micropolar end seed of the lemna, which is called as the operculum. At the end of the micropolar region, a topper-like structure is there in the lemna. So that one we call as the operculum. అది వేరే ఇది గురం కూడా అయితే ఇది అక్కడ ఉన్నది అది ఏమంటారు అంటే మనం కూడా కావాలి గురం కూడా మనం కూడా కాదు తర్వాత మనం గుర్రం కూడా దగ్గర రైట్కి వెళ్ళాలి సార్ కూడానే అందులో ఓపెన్ మెరిట్ అది చూపిస్తే కానీ ఫస్ట్ so this is the stopper-like structure are there in the lemna. That one we call as the operculum. Next one is the carancule. Carancule is formed by the enlargement and the proliferation of the cells and outer integument. Its cells are initially thin-walled, but the latter became a very thick. So, carancule characteristic and the feature of the Euphorbiaceae family. So, in Acalypha, the carancule is found only for the seeds developed from the latter flower of the spike, but not from the lateral flower. So, these carancule, which is helps in the seed dispersal. So, another one is the coma. So, this coma mainly is you can see in the Asclepiaceae family. So, this is the tuft hair of the structures are there, which is the originated from the integumentary tissue near the panicle. These detailed developed for the coma is studied in the Ascleia by the Maheshwari and Lakshmi Narayana. These hairs develop from the epidermal cells of the integument. So these hairs may be grows up to 4 to 5 centimeters in the Caralluma and Karama scenes in the ceramics is also. So another one is the another structure is the obturator. Obturator. So the structure which is generally associated with the function of the directing growth of the pollen tube towards the micropyle are called the obturator. Okay. So which is the shown. So which shows a great variation in the morphology, origin, and anatomy, and the extent of development. So in ovules with the distinct panicle and obturator is also generally funicular origin.
Sir, excuse me. Sir.
Hello? Hello?
Sir, your screen showing only cystiotropous ovule, but you are explaining about endothecium.
Okay. So here, so the obturator here.
Sir, now your screen not showing, sir.
So here, see the embryo sac. So obturator here, you see the in the textbook in page number
No,
here.
sir, not showing. Now only cystiotropous ovule.
Is clear? Hello? Is okay? So this is the obturator to originated from the funicle region only. Okay. So it is the sessile ovule. It is the combination of the placenta and funicle. Okay. So next one is the nucellus. So these nucellus develop from the apex of the ovular primordium. Nucellus is developed from the ovular primordium, and it is the varies in the quantity. It is extent the dimension is also. The ovules has only one nucellus, but rarely in the agile marmalus. Twin nucelli and occur enclosed by the common fold integments also. So this type of the abnormality has been sequenced except in the formation of the false polyembryony. Okay. So sometimes here the archisporial cells undergoes to the transfer division and cut off the outer parietal cells and inner sporogenous cells, sporogenous tissue. So the form by the further division gives rise to the parietal tissue by means which are the sporogenous. So they are became a deep seated on the massive nucellus. So such ovules or sporogenous cells became deeply embedded. So that's known as the crassunucleate ovules. So these are the crassunucleate ovules are we are called. In a few cases, that is the anemone. And sporogenous tissues became the embedded in the nucellar tissues and not by the formation of the parental tissue but the division of the nucellus epidermis. Okay. Such ovules are called as a pseudo-crassunucleate ovules. Of these, tenunucleate ovules are considered the more advanced than the others. So here,
Continue on Managuda Road for 1 kilometer.
సో రాజు కన్నీ తెలుసు. So these cross-nuclear orbital certain units are the advanced that one. So around the cross-nuclear ovals, around the 170 families are observed, but the 10-nucleate ovals are around the 105 ovals are observed. Raju, ఓకేనా?
మీరు అక్కడికి వెళ్ళండి, మీరే
అవసరం డబ్బా. ఆ రోజు ఐఎంఎస్ రాజు తెలియదు.
సంస్కృతి
So these female gametophyte.
bright ఉంటుంది.
The ovule is the source of the female gametophyte and the differentiation of the seed. So here differentiation of the megasporocyst and production of the functional megaspore. So formation of the embryo sac and embryogenesis. So this is all occur within the ovule. So the functional megaspore, after some time, the mitotic gives rise to the small sac-like, that is the sinusotic structure, which is later on organized the female gametophyte. So in generally, the embryo sac are megasporophyte in the 7 cells and 8 nucleate structures are consist of the egg aperture. So here, the embryo sac is consist of the
వస్తున్నారు అక్కడ వస్తున్నారు సార్. వాళ్ళకి రాములు సార్ మరీ చెప్పి పంపించింది కదా.
అది ఇంకా
ఇంటర్నెట్ సార్ని అక్కడ వెళ్ళామన్నారు. మరేమో అది మన ఇది అరవిందో మీరు ఇద్దరు చూసుకోండి అని చెప్పి.
నేను అరవిందులో ఉంటాను సార్ ఇంకా. ఎందుకంటే బ్రైట్ అది.
So here,
చెప్పారు
7-celled and 8 nucleates is consist of the egg aperture and central cells,
ఇదొకటి
and 3 antipodal cells are there.
సంస్కృతి మెరిటి సేమ్ కాబట్టి
The female gametophyte is highly
ఆరు మీ దగ్గర తీసుకురా. నేను అక్కడిని దాంట్లో అరవిందులో
So the female gametophyte is highly సమస్యలు ఉన్నాక So here, so during the development of the female gametophyte, the nutrients required for its growth and development is supplied by the various ovular and the other material tissues are there. నాదేం లేదు నా ఎక్కడైనా దిగుతా ఇక అయిపోయి ఇంకో 15 నిమిషాలు అయితే క్లాస్ క్లోజ్ చేస్తా సో the growth of the female gametophyte cases when it is fully differentiated. The further development of the embryo sac can only seen in the after arrival of the pollen tube on the stigma. The process of the megasporogenesis and formation of the megagametophyte is generally determined. So here, the types of the embryo sac can see the types of the embryo sac here. So depending on the number of megaspores, so megaspores nuclei taking the part of the development of the embryo sac. Here, three main groups of the embryo sacs are developed. So which are the monosporic and bisporic. Another one is the tetrasporic type. So nuclei mainly participating the nucleus are participating in this type. Based on the nucleus are participating the development of the embryo sac. Here monosporic, bisporic and tetrasporic type. So here the first only one of the four megaspores takes part in the development of the gametophyte. So in the second two megaspores, nuclei take in the part of the formation, and the third of the further here subdivisions based on the number of the nuclear division intervening between the time of the megaspore formation and the time of the differentiation of the egg and the total number of nuclei present in the gametophyte at the mature stage. So here monosporic type. In the monosporic type, here the this type of the embryo sac is genetically alike as they are divided by the mitosis of a single nucleus. Again, these monosporic again recognized into polygonum type, and another one is the Enothera type. So this is polygonum type. So Strasburger in 1879 for the first time described this type of the embryo sac in polygonum. The mature embryo sac is in this polygonal type. Here 8 nucleate is there. 8 nucleate is present. So the development of the embryo sac is begins with the elongation of the functional megaspore, which is usually chalagel of the tetrad. From chalagel side of the nuclear is mainly involved in the development of the embryo. The megaspore means the elongation is largely micropolar and chalagel axis. The megaspore cytoplasm is non-vasculate, but the smaller in the vacuoles are appear in the fused together form of the large vacuole. The spindles of the first nuclear division in the megaspore originate along the long axis of the cell. The wall formation does not follow the nuclear division. A large vacuole appear between the two daughter nuclei, result in the form the two daughter nuclei and micropilar and the primary chalagel nuclei. So they are pushed opposite pole sides of the cells. So the most of the cytoplasm is aggregated around them and rest from the thin peripheral layers are there.
నేను అరవిందో అది ఎందుకంటే సారీ పర్టికులర్ గా అరవింద్ అని చెప్పాను కాబట్టి నేను దిగింగ్ ఆడ ఫుల్ టైం వీలైతే ఫుల్ టైం ఉంటాను.
So most of the cytoplasm is aggregated around them. The second division is produced on the pair of the nuclei at each pole, and the third division results in two groups of the nuclei at the opposite pole of the elongated embryo sac. So of the 8 nuclei arising in this manner, the micropilar quarter differentiated into 3-celled egg aperture and upper polar nuclear, and the chalagel quarter into the group of the 3 antipodals. The 2 polar nuclei fuse to give rise to the secondary nuclei. So next one is the Enothera type. So the mature embryo sac is in this type. Here only 4 nucleate. 4 nucleate is present. So which is Gates in 1908 he is reported the first time in Enothera lamarckiana. This type of the embryo sac is usually found in the micropilar megaspore of the tetrad, which is undergoes to the 2 nuclear divisions. So instead of these, usually these 3 are occurring in the polygonum type. Here, the organization of the mature embryo sac, that is the egg aperture and uninuclear central cells. So here, after there is no third division in all these Enothera type of the embryo sac. either the lower polar nuclear or nor any antipodal cells. This type of the embryo sac developed in the form of the characteristic feature of the Anagraceae family. Okay. So next one is the bisporic embryo sac. So bisporic embryo sac. In this type of the embryo sac development, the first meiotic division is accompanied by the wall formation. Okay. So that is the dyad are formed. Wall formation is there, that's why the dyad is formed. Only one of the dyad undergoes the division and the undergoes to other are the degenerated. In the functional dyad, cell wall form does not occur after meiosis 2. So, and both are resulting the megaspore nucleus contribute for the formation of the embryo sac. So each embryo megaspore nuclei undergoes to two mitotic division forming 4 nucleate embryo, and finally organization is similar to the form of the polygonum type.
214.
So normally segregation occurs during the meiosis and or resulting the nuclei. So allium type. So here in the bisporic embryo sac, there are the main two types are there. So in the here two types are there, allium type and Endymion type is there in bisporic type. The allium type, this type of the embryo sac is developed which is first described the allium pistulosum by the Strasburger. So here the megaspore mother cell undergoes to meiotic division first and resulting in the form of the dyad. So of these two dyad cells are chalazal becomes functional while the microphylar degenerated. Okay. So only here the chalazal nuclei is developed as a megaspore. The nucleus of the functional dyad undergoes to meiotic division 2, forming the two nuclei. So these two nuclei move to the opposite poles along the axis and separated by a vacuole. These two nuclei can divide gives to 4 and then after finally 8 nuclei. Botaniaceae and Viscaceae family member. And next one is the Endymion type. So this type of Endymion embryo sac is described in the Endymion. So here the megaspore mother cell undergoes to meiosis 1, forming a dyad. The micropolar dyad is functional, and while the chalazal dyad is the degenerated. So it's the nucleus division to form 2 to 4 nuclei, finally 8 nucleate. 8 nucleate.
ముందుందంటే కదా గుర్తుందా మీరు ఆ కాలేజీకి ఇట్లా పోతూ ముందున్నందుకు నాకు ముందు తెలియదు కాబట్టి సరే మరి చెప్పండి మీరు అదే సార్ కరెక్ట్ చెప్పండి ఒకసారి
సార్ని ఆడ దింపించి లోపలికి ఉంది అది
లాస్ట్ కేసు అంటే కావాలంటే సార్ అదంతా ఉన్నప్పుడు ఒక పావు గంట ముందు నువ్వు పోయి తీసుకుంటావు
రోడ్డు మీద ఉంది కలెక్ట్ చేసుకోవచ్చు.
So it's the nucleus divide and finally 4 nucleates are formed, which is comprising the egg cells and 2 synergies and 2 polars and another one is 3 antipodals. Okay. So as a reduced allium type of the in development is seen in the podostamen and
కావాలంటే లాస్ట్
terminolia.
ముందు సార్ అక్కడ కాలేజ్ దగ్గర ఉన్నా
So another one is the tetrasporic embryo sac here.
అక్కడ ప్యాక్ అయిన తర్వాత వెయిట్ చేయకుండా
ఫస్ట్ మనం ఇక్కడ
So in this group, both meiotic divisions are not accompanied by the cell plate formation. Hence, all the 4 applied nuclei at the end of the meiosis are enclosed in the common cytoplasm formation as the steinomegaspore. So here, the Maheshwari is recognized the 7 principal types of the tetrasporic embryo sacs are namely Adaxa and Peperomia, Pinia, Drusa, Fritillaria, Plumbagella, and Plumbago. So here Adaxa type. It was described for the first time in Johnson in 1879 in Adaxia Mochitellina. So in this type of the embryo sac development, the nuclei of the megaspore takes 2+2 arrangement. 2+2 arrangements here there. All the nuclei of the megaspore undergoes one more division to form the 4 nucleate embryo sac. The mature female gametophyte has a normal egg aperture. Here 3 antipodals and 2 polar nuclei present. So an interesting variation was reported in 400 meters.
Turn left onto Pocharam Road.
So next one is the Peperomia type. So in Peperomian type, here the Campbell and Johnson, they are reported in the Peperomia pellucida. Peperomia pellucida. There are the 2 mitotic divisions in the 4 nuclei of the megaspore after taking up the tetrapolar arrangement and result to forming the 16 nucleate megasporophyte are developed. So here,
Take the next left onto
so finally
Pocharam
organization of the micropolar end form the eggs and synergies. 4 fuse form the secondary nuclei. The remaining 6 are cut off the periphery.
Continue on Pocharam Road for 3 kilometers.
So Campbell also observed the similar type of embryo sac development, and according to him, the egg aperture is invisible 3 cell, approximately 8 nuclei form and secondary nucleus, and the remaining 4 to 6 nuclei are cut off the antipodal cells. So next one is the Pinia type. So in the and sarcocola. Here, the nucleus have the steinomegaspore takes on the tetrapolar arrangement and divide the twice to form 16 nucleate embryo cells. These 16 nucleates are arranged crosswise. Here, 3 nuclei of the each quarter latter cut of cells, while the 4 The remaining three are the moves to the center. So that's why these four tetrad, the polar nuclei are formed. So the central cells of the other tetrad, other triads also simulate the eggs. This type of the embryos are developed in the characteristic family of Phinaceae and some members of the Euphorbiaceae. So another one is the plumbago type. Happened in 1934. Is reported this type of the embryos as plumbago capensis. Undergoes to the another division resulting the four free nuclei are arranged in four pairs. A nucleus of the micropolar pair is now cut off the lenticular egg cells are there. So of the remaining 7 nuclei, 4 are the undergoes to slightly increase the size and gradually approach another one and another the function. So here, the next one is the Fritillaria type. This type of they describe the Fritillaria persica. The cynomegaspore in this type of the embryo type development take up to 1+3 arrangement. The 3 chalazial nuclei are present very close to each other. So during the next division, the micropellar nuclei divides the 3 chalazial spindles fused to form the single common spindle. The 4 applied at the micropolar ends and tripolar nucleus at the chalazial side. So one more division occurs resulting in 8 nuclei. So here.
మూడు కాలేజీలు ఉంటాయి. రెండు కాలేజీలు దూరం దూరం ఉంటాయి. అలా దగ్గర దగ్గర కారు ఉంటే ఒక ఆయన తిరుగుతుంది.
Show the royal